So first, I'd like to thank David and the meeting organizers for inviting me to present at this very special LFS symposium. I mean, it's truly an honor to be here and to be able to interact with the wonderful families and actually the true giants in the P53 world, of which I'm not. As I just noted, I'm a pediatric hematologist oncologist. I currently run the Division of Cancer Predisposition at St. Jude Children's Research Hospital. And, through efforts at our institution and others like Baylor, we've come to learn over recent years that at least 10% of children with cancer develop the disease, due to an underlying genetic predisposition. However, due to the, rarity of childhood cancer and the diversity of cancer predisposition syndromes, we still don't fully understand how the presence of germline mutations influences, clinical presentations and outcomes. And towards this end, I was really fortunate recently to be able to participate in a collaborative research effort with a good colleague at St. Jude, Jun Yang, who sought to examine, the prevalence and spectrum of mutations in P53 in children with acute lymphoblastic leukemia, or ALL. And that's what I'm going to talk to you about today. And although, leukemia is not thought to be a prominent cancer necessarily in LFS, what I hope to convince you of by the end of the presentation is that, germline mutations in this gene do indeed predispose to ALL, where they exert, a very powerful effect. Okay, so I'm a Mac person. What do I do to make it go forward? There we go. So for those who are less familiar, childhood ALL is really the most common pediatric cancer. It comprises about 1/3 of all new childhood cancer diagnoses. Fortunately, due to advances in treatment, for the most part, this cancer is highly curable with contemporary risk-adapted, multi-agent chemotherapy approaches. And thanks to the work of, many people in the field, there's now growing evidence for a role for inherited susceptibility in childhood ALL. So this has come through genome-wide association studies that have shown that, variants in or around genes such as, CEBPE, CDKN2A, ARID5B, GATA3, and actually family-based studies that have demonstrated truly, damaging germline mutations in genes such as PAX5, ETV6, and IKZF1, all play some role in the development of childhood ALL. And kind of relevant to today's presentation, Linda Holmfeld, who works in the laboratory of, again, another colleague at St. Jude, Charles Mulligan, she made a very important observation that she published in 2013, which is that up to 50% of children with a rare form of ALL, known as hypodiploid ALL, carry germline mutations in TP53. So these children have Li-Fraumeni syndrome. So hypodiploid ALL is a little different from the other forms of ALL in that the number of chromosomes in the leukemia cells is lower than what you normally see, just so you know what it is. So normal human cells have 46 chromosomes. When you have hypodiploid ALL, the leukemia cells have somewhere between 32 and 39 chromosomes. And importantly, compared to the more common and traditional forms of ALL, where cure rates now hover around 90%. Children with hypodiploid ALL, have a much poorer prognosis. So from a paper published in 2015 by Dr. Mulligan and Dr. Pui at St. Jude, you can appreciate here that patients with hypodiploid ALL have a, estimated survival of about 70%. However, that survival can vary greatly depending upon whether there is evidence of leukemia circulating still at the end of the first phase of induction, or therapy. This is known as induction. So circulating leukemia is also called minimal residual disease. So for those patients with hypodiploid ALL that have minimal residual disease at the end of induction, you can appreciate that survival can be as low as 20 to 30%. So, these observations kind of set the stage that P53 may play an important role in the development of this rare form of childhood leukemia. But the question remained, what is the extent to which germline mutations contribute more broadly to the development of childhood ALL? And from my perspective as a clinician, how does the presence of these mutations impact necessarily presentation? So is disease more advanced? Or outcome? Do these patients respond to therapy? So if you think about it here, if we know that 50% of these patients carry P53 mutations, where do they fall on these curves? And that wasn't known at the time that this paper was published. So To begin to address this question, Jun Yang and a very talented postdoc in his lab, Max Jian, set out to complete a sequencing study, a targeted sequencing study using germline DNA samples, and these samples came from peripheral blood or bone marrow that was collected while children were in remission, so technically the leukemia wasn't obvious in those tissues, and these children were recruited from 2 large frontline ALL trials run by a large, pediatric oncology consortium, the Children's Oncology Group. So these children were recruited from the high-risk BALL study, AALL0232, and the standard-risk high-risk ALL study, P9900. So this is a study that started in 2002. This one kind of predated it. Patients were excluded from the study, obviously, if they didn't have any germline DNA, if targeted sequencing failed, or if there was insufficient clinical information. So at the end of that, about 3,800 actually patients were enrolled in this sequencing study, and this actually represents about 75% of all the patients on those 2 studies. So it's a pretty significant number. So what Max did was, he completed capture-based NGS sequencing of the TP53 locus in these children, aiming for at least what's called a 20-fold coverage of depth across 99% of the P53 gene, okay? The sequences that were obtained were aligned and they were mapped back to the human genome, and then there's a program that you can use to look for variants called GATK. This is a very common kind of variant calling pipeline, and so he used that to identify alterations in the gene in patients with ALL. And ultimately, what Max identified was that there were 49 very rare, unique, what we call non-silent. So these are changes in the gene that lead to alterations in the amino acid sequence, P53 variants, that were present in 77 patients. So this is about 2% of that total cohort. And these, for the scientists in the room, 40 missense mutations, 1 nonsense, 6 frame shift, 2 in-frame deletions. Now, this is all fine and good, but were these variants really critical variants that could be considered pathogenic or disease-associated? And there are ways that you can look at these genetic alterations to try to get an idea. So these can be annotated, it's called, based on what type of a variant they are, and then by comparing the information that you get to information that's present in online databases. And there are some common databases we use, a very powerful one run in France, the IARC TP53 database, and then there's another excellent NIH-funded project, ClinVar, and then there are computational algorithms that you can use. So we use programs called SIF, Polyphen, and we use a CAD scoring system. And so in this scenario, basically, if you have a rare variant and it's a non-sector of frame shift, this kind of cuts the gene so that it makes a short protein, that would automatically be considered a pathogenic or a disease-associated variant. If that were not the case, so if you had a missense, a nucleotide change, and you went to the IARC database and it was considered non-functional in one of their functional assays, a transcriptional assay, that would be considered pathogenic. If you had a missense change that actually was considered functional, so it retained P53 function in a transcriptional assay, those were classified in this study as variants of uncertain clinical significance. So these are nucleotide changes, but they appear to still function. And then if you had variants that actually you go to the P53 IARC database and you find that, there's a little bit of function in the transcriptional assay, we would then go to the computational algorithms, and if they were damaging across the 3 platforms, they would be considered pathogenic, and if they were not, they were considered variants of uncertain significance. So this is kind of the algorithm that was used for this paper. In many ways, it kind of mirrors what we now use in the American College of Medical Genetics and Genomics, and I kind of checked before I came, and most of the variants that were reported in this paper would have been classified similarly using current clinical guidelines. So at the end result of all of this, of the 49 variants initially identified, 22 were classified as pathogenic or disease-associated. So that's 6 frame shifts shown in green, 1 nonsense shown in red, and 15 missense mutations. 11 of these mutations, or 50%, had not been previously described in the IARC P53 database as germline variants. 14 of the 15 missense clustered in the DNA binding domain. This is very consistent with what we know about these germline mutations in P53. And actually, so 9 of them, clustered here in a region that's important with either binding to DNA or in maintaining the kind of physical structure of the DNA binding surface of p53. To determine whether, pathogenic p53 mutations were enriched in this big cohort of ALL patients compared to control individuals, this is another way that you can tell that these might be associated with the development of the disease. Max carried out a very similar analysis where he actually screened genomic sequences that are now available publicly from a large control cohort of individuals who theoretically do not have ALL. He identified variants, he put them through that classification pipeline. And in this comparative study, he found that there were 43 variants identified in 81 participants for a prevalence of 0.1%. Okay? And if you compare the prevalence of p53 mutations in the ALL cohort and in the non-ALL cohort, what he found was that there was a statistically significant overrepresentation of the p53 variants in the ALL cohort, with an odds ratio of 5.2 and a p-value of 4.8 to 10 to the minus 10, suggesting that yes, indeed, these p53 variants are very likely related to the development of ALL. Now, that's important to compare to the variants of unknown significance that were identified, where actually there was no enrichment. So VUSs, we don't usually think impact the function of the protein. As clinicians, we often don't act upon them clinically in change management, and this would support that perhaps in this study they really are not associated with ALL risk, at least significantly. So now comes the part that we want to get to, which is, how do these variants relate to clinical presentation and outcome? So when Max looked at clinical variables that are available through the COG, data repository, he found, which I can't really explain, that patients with pathogenic mutations tended to be older than patients with wild-type p53 or variants of uncertain clinical significance. Patients tended to have a white blood cell count less than 50,000. Usually when it's over 50,000, these patients are considered as higher risk. Less than 50,000 is considered more treatable ALL. Consistent with the work of Holmfeld earlier, he found that, the large proportion of patients with germline variants had this type of ALL called hypodiploid ALL. But I think it's important to note that, germline p53 mutations were not restricted to that cohort. So, when I read Linda's paper originally, I thought, oh, well, you're only going to find p53 in kids with hypodiploid ALL. But that is not the case. About 35% of patients with germline mutations have the more traditional forms of ALL, hyperdiploid or diploid. So we have to remember as clinicians that this can still pop up in appropriate patients with more traditional forms of ALL. After completing his analysis, Max did find no association of the variants with other features of the disease, such as gender, genetically defined race, presence or absence of MRD at the end of induction. Remember, we go back to that first graph. So I had assumed going into this that the p53 positive patients would be the ones with the with a positive MRD and perhaps poorer survival, but that wasn't the case. And the mutations were not associated with any of the more traditional leukemia-defining chromosomal translocations. So there's something called ETV6 RUNX1. There's a Philadelphia chromosome. There really is no association with germline mutations in any of the kind of classic other chromosomal abnormalities we see in childhood leukemia. So now in terms of outcome, and this is where the story gets a little bit more depressing. So if you look at the entire, AALL0232 cohort, so all comers, what Max observed was that patients with a p53 pathogenic variant, they tended to have a significantly worse event-free survival and overall survival compared to those who had wild-type p53 or a p53 variant of uncertain significance, with a hazard ratio of 2.8 and 3.1, respectively. And this is all patients. So how much do the hypodiploid patients, contribute to this? I'm not going to show you the slides, but if you draw these same curves and you focus right in on that 65% that had pathogenic, if you focus in strictly on the hypodiploid patients, you actually find that there's no difference in either of these curves. So having hypodiploid ALL kind of negates the negative prognostic impact of a germline p53 mutation. Well, what if you look at those other patients, the ones that don't have hypodiploid? So if you exclude hypodiploid ALL, you again see that having a p53 mutation confers a poorer prognosis in children with ALL. now with higher hazard ratios of 5.4 and 6.1. And I don't show you here, but basically data from the 9900 study showed exactly the same thing. And this kind of mirrors, I think, what we all felt as clinicians, but now we have, more definitive proof with a larger number of patients. So, what's going on? Why are patients with germline mutations? Oh, sorry, before I get to that. This just goes to show you that in children with variants of uncertain significance, if you compare their outcomes to those with wild-type P53, there is no statistical difference. And that goes along with kind of what we showed earlier, that there's no statistical enrichment of VUSs in ALL patients versus control. So the VUSs don't seem to have a clinical impact. Okay, so why? Why are these patients tending to have a poorer outcome compared to patients with wild-type P53 or variants of uncertain significance? So as clinicians or oncologists, we all know that, relapse is a big problem in children with leukemia. Well, not a big problem, but when it occurs, it's a problem. So it generally occurs in about 15-20% of leukemia patients across the board. But you can appreciate here that, a good proportion of patients develop relapse regardless of whether or not they had a germline P53 variant. That's shown in blue. But what catches the eye here is that, a much larger proportion of patients with P53 pathogenic variants develop second cancers compared to those with wild-type P53 or variants of uncertain significance. And this equates actually to cumulative incidence, of second cancers of about 25%, compared to only 1% for patients with wild-type P53 or P53 VUS. And if you look at the kinds of second cancers, and numbers are relatively small, but what you see here are 2 cases of myelodysplastic syndrome. So this is treatment-associated MDS. These are patients that did not receive radiation, but received bone marrow transplantation as part of the treatment of their leukemia. 1 leiomyosarcoma in a patient that got total body irradiation in the setting of a transplant. A malignant teratoma, and then a malignant glioma that occurred in a patient that had both a transplant and a radiation. So second cancers are a problem. This gets back to what Louise just mentioned. Second cancers are a big risk, and actually they occur relatively early in these patients compared to the general population of children with cancer. So with that, I will summarize that through this study, Max was able to identify 22 unique P53 coding variants in 26 of his 3,800 patients. This is about 7.7% of patients. And while this may not sound like a big number, up to now it was thought that maybe a small proportion of children with leukemia harbored a predisposition. And I'm going to throw out a number, but that comes kind of from thin air unless Josh and David have better ideas. But, I'd heard previously maybe 3 to 5% of patients with leukemia have a predisposition. It may be higher than that. But, if you think that it's that low, this could be, almost 20 to 25% of all patients. P53 variants are significantly overrepresented in ALL patients compared to controls. Carrying a pathogenic mutation, unfortunately, is associated with a poorer outcome. And children with P53 variants are at a significantly higher risk to develop second cancers, with a 5-year incidence of 25 versus 1%. So the question remains, really, where do we go from here? How can we improve upon our current information? So how do we best treat ALL patients with P53 mutations to minimize the risk for relapse and second neoplasms? What did I put here? Oh, didn't put anything. So one thought that I had as I was sitting up front trying to think about new thoughts was, this is an amazing time in terms of immunotherapy. I came from Children's Hospital of Philadelphia, where I was very lucky that they had developed this new program for the families of what's called chimeric antigen receptor, so modified T cells, which are immune cells that can actually attack your leukemia cells, and they can eradicate your leukemia cells in a very effective way. And while this would be very atypical, but in the future, could we use an immune-based therapy to treat ALL as opposed to a toxic chemotherapy that we know induces DNA damage? We try to avoid as best as possible radiation therapy. And actually, I do know that this T-cell therapy can be used in patients with LFS. You can actually take T-cells out and engineer them and use them to kill cancer, because my colleague at CHOP has treated a patient with relapsed LFS, an LFS patient with relapsed leukemia with these engineered T-cells. What can be done to predict when and in which patients a second malignant neoplasm is most likely to occur. So are there certain genetic modifiers, certain p53 genotypes and methylation patterns? Are there specific treatment exposures we can avoid? And ultimately, what can we do to detect these second neoplasms, hopefully earlier, when they are most likely to be cured? And that will lead to some nice discussion later about circulating tumor DNA. So with that, I will close. I very much want to thank, all the families and patients at St. Jude that have served as my inspiration every day for what I do. And then I have to give thanks to Jun and Max at St. Jude and all of their collaborators at COG, because the study would not have been possible without them. And then my wonderful cancer predisposition team, I have to show you guys. For Halloween last year, we were the genomes. Get it? Art, 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 art. That's me in the middle. And if you need to reach me, it's GP team, genetic predisposition team at stjude.org. All right, so I'm going to stop. Thank you very much. Yes.
That was fantastic. So I presume that the tumors lose the other allele of P53?
Yeah, I'm sorry. So in this study, June didn't sequence the tumors, but he has since requested tumors from those individuals who had germline mutations, and he's in the process of looking. So, in the few cases that we've sequenced clinically, we do see, in some cases, mutations in the second allele, but I can't give you a really strong answer yet because we've only had rare clinical cases, and he'll be looking in more depth.
And also, this is just in BALL, not TALL.
This is BALL strictly, no TALL.
No TALLs are ever seen with...
I don't want to say that. It's just these were clinical studies restricted to B lymphoblastic leukemia. Okay,
Great, thank you.
Yeah. Josh?
I'll just shout. So thanks for a really great talk.
Oh, did somebody not shut theirs off?
Oh.
Okay. No. Josh, you can just holler. Josh,
yeah.
you can't holler because they can't hear you. Oh, well. I can speak it. I can repeat it.
So thanks for a great talk, very clear. So I think you've shown convincingly, and your colleagues, Gene, everyone, so about 1%, a little less than 1% of leukemia is due to these germline P53 mutations. Do you have a sense of the converse? So we always say 3% or so a risk of developing hematological malignancies in LFS. I assume we don't have parents to know if those are de novo or not.
Oh, I wish I did.
I'm just wondering, what do you think, as the expert, of what percentage is truly Li-Fraumeni patients
Develop leukemia. Oh, I don't know off the top of my head because we didn't look this way, but these are important questions to answer. I mean, David, you have a much bigger cohort. I don't know, Louise, if you'd be able to answer that more specifically. I can't tell you off the top of my head from this study because parents weren't sequenced, and we don't have a big enough cohort yet at St. Jude to answer that. David?
I don't know that we have enough patients with leukemia in our cohort to answer it, I think. I have another question.
Oh, go ahead. Sorry, Josh, so I can't answer your question. I think it's rare in LFS, but I think it happens. And when it does happen, it's a problem. Yeah.
Very quickly, so the survival data is intriguing, but it doesn't, not that we necessarily know for sure, but for other cancers in the context of germline P53 mutations, we don't necessarily see a poor survival advantage. I mean, some would argue we actually see improved survival because patients actually go on to develop multiple malignancies. So you have any sense of why there might be something in the ALL context?
Well, I wonder if so David's asking, or basically survival may not necessarily be bad or worse, in individuals with other tumor types that harbor germline P53 mutations. Why might ALL be different? I mean, I don't have the answer. I imagine in some of these other cohorts, chemotherapy is equally intense. In leukemia, I know it's quite prolonged. I wonder if it has to do with kind of the cumulative impact. Now, you have to remember, too, some of these patients, these clinical trials, 0232 is a little bit more current, but it's still relatively old. 9900, I have to go back and look at the protocol. I don't know how routine radiation was as part of that protocol. That could have been a big part of this, right? So as therapy evolves, that outcome may change. That's our hope, right? But I don't have a specific answer off the top of my head.